Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Todd M. Brusko. He's an associate professor with the Diabetes Institute at University of Florida in the College of Medicine, and he appears to focus on type 1 diabetes. So we'll talk about that. So Todd, thanks for coming.
Welcome. Thank you for having me.
Yeah. Usually the people that study type 1 have type 1 or know someone that does have it, but what's your reasoning why diabetes and why this type?
Yeah, no, it's a great question. So I've had family members with type 2 diabetes, but not type 1 specifically. So type 1 diabetes is, it fits in this category of being autoimmune in origin. And I've always been fascinated with the immune system in general and the complexity of it, including, some of the genetic origins of the disease itself. So it started for me back, all the way from a time when I was a young undergraduate researcher in a genetics of type 1 diabetes lab, and I've just stayed with it ever since.
Okay. Well, what aspects of type 1 are you focused in on?
Yeah, so my lab does a mixture of different research. So most of what we do is trying to understand how the disease forms in individuals that are at higher genetic risk. So individuals that have family members that may have type 1 diabetes as well. So from that perspective, we try and understand what genes are involved in the disease process and how those influence how the immune system responds in individuals that are at higher genetic risk. And then we're interested in biomarkers and as well. So as people grow and develop some of the biomarkers of disease progression, the most apparent are what are referred to as autoantibodies. So individuals eventually develop antibodies that recognize parts of their own beta cells that make insulin. But there are also other biomarkers. So it's primarily a T cell driven autoimmune disease. So we're interested in tracking how T cells develop over time and working on developing biomarkers in that regard as well. My lab also has a strong interest in regulatory T cells. So these are a small white blood cell in circulation that is there to keep the immune system in check. Actually, if you have a mutation in one of the key genes that is responsible for Treg development, you develop not only diseases like type 1 diabetes, but really severe forms of autoimmune conditions that attack all sorts of organs throughout your body, this syndrome called IPEX syndrome. So we've been interested in a long time in trying to correct some of these defects in people that develop autoimmunity and finding ways to do that in a much more targeted and sort of precision medicine guided strategy. And so that involves understanding the different paths that people take to disease and also trying to target specific antigens and these T cells that we think are important for keeping things in check.
Are people born with type 1? And, why is there a latency? It seems like there's a huge variability in
that is an amazing
when they get it.
Variability. Yeah. So we're finding some of the earliest biomarkers of disease progression in kids as young as 6 months to a year of age. We can begin to find some of these autoantibodies. So the implication there is that some of whatever is triggering the autoimmunity to start can happen very, very early in life and potentially even in utero in some of the subjects that are at the very highest genetic risk for developing it. On the other end of the spectrum, you can develop type 1 diabetes all the way out to 80 years of age. So even though we used to call it juvenile form of diabetes, it really can develop through the entire lifespan. So it's really variable in terms of when it can actually present itself.
Typically, when's typical? Yeah.
Yeah, so typically, I mean, you see this sort of peaking right before puberty in most subjects locally here, where we see our largest peak of disease incidence. But when we, like I said, when we follow some of these serological biomarkers, these autoantibodies, they show up oftentimes within the first 2 years of life, and it may just be a longer drawn out process for when the disease actually clinically presents. So these kids oftentimes they'll present with being extremely thirsty and constant urination, and they'll, in the most severe form, they actually go into a state called diabetic ketoacidosis, where they oftentimes wind up in the emergency room and need to get their blood sugars back into the normal range. So the clinical presentation can be quite severe, and that's really one of the reasons why we're so interested in finding these biomarkers of disease progression.
Well, what are some of these specific antibodies, and what do they tell you? What are the antibodies against?
Yeah, so these antibodies are really they're markers. They tend to recognize parts of a beta cell itself. So they're responsive to insulin or forms of insulin. There are other proteins that are present within beta cells, these cells that make insulin in your body, called GAD, IA2, ZNT8. So these are all things that are kind of tissue restricted, and they really point us to this targeted autoimmune response that's occurring in patients that develop type 1 diabetes. And the thing that is really powerful about them as biomarkers is that the more you have, the more likely an individual is to develop disease. So once an individual has 2 of these autoantibodies present, they have a fairly high chance over the next 5 years of developing disease.
Has anyone identified how type 1 starts?
Yeah, so there's large groups and consortia throughout the U.S. and even throughout the world that are looking at this much more difficult question of what are some of the environmental triggers of diabetes. So clearly there's been a long-term interest in viral infections as a potential trigger for breaking what we refer to as immunological tolerance. What happens in autoimmunity in general is that your immune system should tolerate your own tissues and not fight your own tissues as being foreign. And something that occurs, whether it's because of underlying genetic susceptibility or exposure to something like a viral trigger that causes a break in those normal defenses, and this trigger of autoimmune destruction. So there are hints that there are repeated viral infections in early development. In type 1 diabetes, there have been some evidence of enteroviral infections being involved in the early stages of development of the immune system as being linked with the progression of autoimmunity. So certainly there are hints there. What we haven't seemed to really hone in on are viruses that would directly infect beta cells. And there's been a lot of extensive research looking for those types of viruses that might cause direct infection within the pancreas and within the islets that hold these insulin-producing beta cells.
Yeah, but so far as I know, the pancreas has like 2 functions, endocrine and exocrine function. But type 1 diabetes appears to be very specific to beta cell destruction only, and not the other pancreatic cells. So I guess it's either like the membrane expression or the products produced by the beta cells.
So I think we're learning more and more as we go along here. So I'm involved in this large network called the Network for Pancreatic Organ Donors with Diabetes that's located here at the University of Florida. And basically what we do is if anyone dies in the continental United States and they happen to have type 1 diabetes or these biomarkers of type 1 diabetes, and they become an organ donor, we actually try to acquire their pancreas locally here at UF, and we process that pancreas and then distribute it to investigators throughout the world to really understand what's actually happening in that target organ itself. So that then goes out to over 150 affiliated researchers throughout the world. And one of the things that we found is that it does not appear to be just the islet and just beta cells that are impacted. So the pancreas in general may be as much as 45% smaller in someone that develops type 1 diabetes. So that can't be explained simply because of the loss of beta cells that make insulin. So there's some other inflammation or atrophy going on in patients that develop type 1 diabetes. And we actually see this in some of the relatives as well. So, or those that are at risk for progressing to disease. And this has been shown now by MRI imaging that the pancreas is just generally smaller. So, perhaps some of these individuals kind of start off with less overall pancreatic mass or beta cell mass, and that puts them at higher susceptibility for developing disease. So there's subclinical deficiencies of some of these other acinar functions as well. So there's less trypsinogen and lipase that are being made out of the exocrine pancreas as well.
But if the pancreas is smaller, they have less of an endowment of beta cells. Why wouldn't they fall into type 2 more readily versus type 1? Why would there be an autoimmune response instead of just again, type 2 response?
Yeah, so that's a good question. So the path down the type 2 versus the type 1, sort of the disease presentations are very different, and they're very different in terms of genetic susceptibility as well. So the genetics of type 1 diabetes become very complex. And without getting into too much detail, all I will say is that the majority of the genetic risk, so almost half of the overall genetic risk for type 1 diabetes, comes from this one region in the genome around chromosome 6 that encodes something called HLA, or human lymphocyte antigen. And that's common, a common gene that's often associated with autoimmune diseases in general. And that's so the genetic risk elements in type 1 diabetes are these molecules called primarily DR3 and DR4. Now, type 2 diabetes is a very different clinical presentation. So it's not much, it's not as associated with autoimmunity as it is with insulin resistance overall. So the whole clinical presentation is very different, but type 2 diabetes does have a somewhat reduced pancreatic volume as well.
Oh, really? Huh.
Yeah.
So is it that, do you think people are born with it when they get type 2 diabetes that predisposes them?
Yeah, so it is a good question. So at least in type 1 diabetes, my colleagues and collaborators, Dr. Michael Haller and Martha Campbell Thompson, are doing longitudinal imaging studies of individuals that are at elevated risk. So relatives of type 1s and subjects that have these multiple autoantibodies present. And it does appear that during the disease progression, there is some shrinking of the pancreas on the way to the development of type 1 diabetes. So that it may be that there's a lower starting point, but there's also clearly progressive loss of mass as well as individuals progress.
I guess, you can't really biopsy a pancreas, so it would probably be dangerous and super invasive.
Yeah, no, that's a great question. And that's been tried actually. And it was just as you suspected, it was kind of a bad outcome in terms of what you could imagine from poking a hole in the pancreas that's full of digestive enzymes. So that will likely never happen again. So if we're going to do these types of longitudinal studies, it's going to have to be from these non-invasive imaging types of approaches where the pancreas itself is not disturbed.
Of the cadaver pancreases that have been obtained, is there a wide age distribution?
The program in general has collected over 500 pancreases from organ donors, and that includes, not only patients with type 1 diabetes, but control pancreases and pancreases from individuals that may have other rare forms of diabetes. So monogenic forms of diabetes from mutations in key genes for insulin processing to CF-related diabetes, cystic fibrosis-related diabetes. So there's a lot of different comparisons there to really understand what's unique about the pathology in type 1 diabetes versus some of these other clinical presentations that are present in other forms of diabetes as well. And they span the whole age range from very young subjects to what we refer to are for studies that are titled the medalists that have had type 1 diabetes for decades. And one of the fascinating findings out of NPOD is that even in subjects that have had type 1 diabetes for decades, we can still find some residual islets and beta cells that contain some insulin often. So it's not a complete loss, and there's some hope that we could somehow recover or restore some of that endogenous beta cell function in even individuals that have long-term type 1 diabetes.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
What about using stem cells or induced pluripotent stem cells and differentiating them to be islets and then implanting them back into a person with type 1?
Yeah, so there are a number of groups that have those types of approaches underway. So that's kind of a restorative type of therapy. So if someone really has destroyed so much of their endogenous beta cell mass that they can no longer produce sufficient insulin, then you have to think about those types of therapies. So it could be from IPS, and I think that has a lot of advantages in terms of being the same genetic sort of copy as the person who might be receiving it, or at least matched in terms of those tissue matching antigens that I was talking about as well, or these embryological ES-derived stem cells as well. There, I think the challenge becomes, how do you then protect those derived beta cells from not only, so if they're not genetically matched, you have to protect them from alloimmune responses. So someone's immune system will recognize them as being foreign and potentially kill them off. Or in the real challenge in type 1 diabetes as well is that because they developed autoimmunity initially, you not only have to protect them from that alloimmune response, but you have to protect them from an recurrent autoimmune response. So because these are, going to be new islet or insulin producing cells, they have the potential to again reject them from that autoimmune response. So there's a couple challenges there, and there there are groups of investigators that are trying to put them in protective devices or cover them, kind of sheathe them from recognition by the immune system. And prevent them from being rejected again.
Is there any way to look at the blood, the downstream blood from the islets, and look for like extracellular vesicles in it that they can tie back to the islets? There's
a lot of researchers who have been looking at all of these signatures of what is a beta cell and what does it release into circulation, whether it's microRNAs, these molecular biomarkers of unique cell subsets, or the vesicles that you're talking about. So there's certainly lots of efforts to, and this is really one of the key needs in the field, is to how do we monitor the health and survival and numbers of beta cells in individuals as they progress to type 1 diabetes or after a replacement therapy like transplanting these stem cell derived beta cells.
Is there any way to put any some kind of an implant into the duct, the pancreas? I know you wouldn't get the beta cell function that goes in the blood, but, maybe by looking at the other function, you can infer what's going on in the beta cell side.
Yeah, so people have looked at differences in sort of where these replacement therapies would go. And the determination of where it will go is a combination of what is safe from an FDA perspective in terms of if something were to go wrong, can you easily access it and retrieve that type of material? And then also there's this consideration of where is it going to be sufficiently vascularized so that it engrafts well and functions well. And there are also some differences in where it may reside in terms of how the immune system may actually respond to that engraftment as well. So all of these are ongoing areas of active research, including this notion of encapsulating these new beta cells and then protecting them from rejection by the immune system. And, it's not just a physical protection. The exciting thing that we're able to do now in terms of gene editing some of these cells at an ES stage or even in an IPS stage is that we can think about knocking out genes that the immune system needs to kind of recognize these tissues And that obviously presents its own set of challenges, but it is an exciting time to think about shielding them, even by genetic editing, from autoimmune attack.
So the autoimmune attack, what is the nature of it? You mentioned T cells earlier on. I've heard other people talk about that. So has it been specified, the method of attack?
One of the clearest signals that we have of what is going on is when we look in these organ donors with type 1 diabetes, and we look in histological sections of the pancreas. And when we zoom down in on islets and see beta cells at disease onset, sort of, when the disease is most active, we can find these infiltrating white blood cells, lymphocytes, right there in the islets, in proximity to insulin producing beta cells. So we know that these beta cells have their flags up, that something is going on, that there's some sort of stress in that local microenvironment. And that puts them at sort of in the state of being surveyed by the immune system. And we have white blood cells, CD8 cells in particular, that can directly recognize antigens in beta cells. And the thinking is that those CD8 T cells can directly kill or lyse beta cells. So there have been a number of clinical trials in patients with type 1 diabetes to target the immune system and block some of that autoimmune T cell killing. And the agents that have had some of the most success in the clinic are agents that recognize antigens that are expressed on the surface of T cells. So anti-CD3. CD3 is this molecule that's associated with the T cell receptor. Alefacept, which recognize a molecule that's on the surface of activated effector cells as well. And then here at the University of Florida, we've been working with anti-thymocyte globulin, which is a combination of antibodies that recognize primarily T cells as well, and can blunt some of the host immune response in individuals that are developing type 1 diabetes. So all of those agents have shown some efficacy in blocking some of the autoimmune attack in individuals when they present with clinical onset of disease. Anti-CD3 has actually been moved back in that process to the point where it's been patients that show those autoantibodies that we know are at really high risk of developing disease have been given anti-CD3, and it's been shown to delay the onset of disease for as much as 2 years in some subjects. So there's a hope that as we understand more and more about how this disease occurs, that we'll have a better sense of how to target some of these cells that are doing the killing, and hopefully, re-educate them towards what we refer to as immunological tolerance over time.
Well, what are the beta cells expressing on their membranes that's different? That would, if you put them side by side, a beta cell that is open to immune attack versus one that's not, what's the difference?
Yeah, it's a great question. I think it comes down to how much stress they may be under, metabolic stress, and how they deal with that stress. There is some evidence that the way a metabolically stressed beta cell, the types of proteins and things that they present on their surface change over time under states of inflammation or inflammatory stress. And a lot of this comes down to that question of, is it the genetics that's putting someone at increased susceptibility, or is it these environmental stressors or viral infections that's putting them at increased susceptibility, or is it the combination of the two? And that's really where the complexity of it falls out. So it's not just these HLA molecules that put someone at higher risk. There are about roughly 60 other loci in the genome that confer very smaller amounts of overall genetic risk. But we can kind of get a sense for what that is by adding up all of these other minor risk loci throughout the genome.
Well, what kinds of items are expressed on the membranes of the cell?
So there are pieces of insulin that wind up getting presented on the cell surface. So every cell in your body has kind of a machinery to take little pieces of itself and present it on the surface for the immune system to come around and surveil. And normally your immune system doesn't respond to those alone if you're not in a state of active infection. So if you can think about a scenario where if you got infected with a virus and that virus got into your cells and used your cellular machinery to make additional copies of that virus, then your immune system kind of gets a sense that something is going on and it has to kill off those cells to prevent viral spread and replication. So it may very well be that the immune system simply thinks that there is an ongoing stress or viral infection in beta cells of individuals that develop type 1 diabetes. Even if there may not be, there's probably signals that are being received in that cell from stress that are putting up these flags for the immune system and these CD8 T cells to recognize them and ultimately wind up killing them.
Well, I mean, what would stress a beta cell? I guess its job is to produce insulin. So if there's fewer of them, if the pancreas is, let's say, half, or the endowment of beta cells is half, that would probably put them under a lot of stress to do double the work. Let's say.
Exactly. Yeah, no, so you're asking some really insightful questions about what is causing this stress. And beta cells are under an exceptional amount of metabolic strain in terms of turning out so much insulin that it actually packs into granules inside beta cells. And some of our current thinking is that as you have to make a ton of this insulin within a beta cell, that also requires that there's a lot of messenger RNA that might build up and not be effectively translated into protein. And cells in your body have this unique ability to sense cellular stress and too much RNA building up. And that can raise some red flags for the immune system as well. So just by sheer metabolic strain and stress, it may actually put up some of these flags for additional immune surveillance. These are probably events that are occurring in all of us. So it's still this key question is why does the immune system of individuals who develop disease handle it in a way that creates this long-term downward spiral and loss of additional beta cells? And that is sort of a key question that is not fully addressed in the field.
There's 500 pancreases. Do they have different morphology? I mean, you said they're smaller. And then what about the percentage mix or the distribution of beta cells versus other cell types in them? Yeah,
so those studies are underway now by all kinds of exciting new technologies. So we can now go in and at the single cell sort of resolution, we can understand what genes are being expressed in beta cells versus alpha cells or delta cells within islets. It does appear that this process is not uniform across the entire pancreas, and that, if anything, it seems to be enriched in the tail of the pancreas, kind of proximal to where it connects to the spleen, and then it may spread upwards towards the head of the pancreas. It also just happens that that region has more insulin-producing beta cells, so there may just be more antigen there to kind of drive this process. But it is very variable in terms of what's happening, not only between different individuals, but also you can have two islets almost right next to each other. One will be completely destroyed and the other will still look like a normal sort of islet and may actually still have insulin producing beta cells. And I think one of the interesting questions that we think about is that you would expect your immune system to target and kill off all of those beta cells, and it doesn't. So what is it about those islets that is different? What is it about whether it's vasculature or innervation or something that is keeping them protected from autoimmune attack?
Yeah, that's another question. Are there any intermediate forms? So I've heard when they look and they say the beta cells are gone, they're just literally gone. There's no scarring. It's like they were never there. So are there any intermediate forms in these cadaver pancreases where you see certain islets that are halfway through destruction, or do you only see like perfectly good ones and non-existent ones?
No, there's definitely intermediate forms. And it also gets back to that notion of heterogeneity and disease progression. So in general, and I say these as general concepts, but the younger individuals are that develop autoimmunity, the more severe the clinical presentation often is, and the more complete the loss of beta cells are. So if you develop type 1 diabetes at the age of 2, you have a fairly strong autoimmune response, and the loss of beta cells seems to be quite rapid. And that's very different than if you develop type 1 diabetes at the age of 21, for example, where you have this much slower loss of beta cells and clinical presentation. And oftentimes in those individuals that develop disease later in life, that's where we'll see these residual islets and residual beta cells more often than in some of the individuals that have developed at a very young age. So there's quite a bit. It's very closely linked to age of onset, but also some of those genetics that seem to be playing a major role.
Well, do you get any information by studying these intermediate forms of islets? Like can you now gauge destruction of an islet? What happens first? What happens near the end?
Yeah, it's a great question. And, one of the ideas that is being investigated by individuals now is whether there are protective proteins that are being expressed on the surface of some of these beta cells that still have residual insulin. And one of the molecules that comes to mind is this PDL1 molecule that might get upregulated on beta cells. And one of the reasons we think this may be an important protective mechanism is that individuals that develop cancer and are receiving these immune checkpoint inhibitors, a small population of them are actually developing really rapid onset type 1 diabetes, implying that if you block these checkpoints for the immune system, it's almost releasing their immune system to recognize some of their own tissues very rapidly and induce autoimmunity. So that's one of the theories that is being investigated now about why you might have a protected islet versus an islet that's fully destroyed by the autoimmune response.
Last thing to ask you is the signaling to tell the pancreas, hey, produce these enzymes, produce insulin, etc. Does there appear to be an over signaling or preferential signaling for more insulin versus other pancreatic enzymes, or is the signaling altogether somewhat corrupted or overactive or underactive in these patients? Can you tell?
So what we've seen from studies within NPOD and we've seen some biomarkers in circulation as well, is that insulin itself comes in this pre-pro form, and it has to be processed into mature insulin. And the processing machinery seems to be dysregulated in individuals that develop type 1 diabetes. So you can kind of think of an assembly chain, and you have these molecules of insulin that are coming down the pipeline. And if you speed that chain up too fast and you make mistakes in the way that insulin is processed, you kind of gum up the works, and it creates some cellular stress as well. And that may make them more susceptible to autoimmune attack. So there are interesting observations that are being made in individuals that develop type 1 diabetes, that they lack that processing machinery where you cut out this C-peptide portion of insulin and you have more of this pre-proinsulin that is just kind of residing in beta cells in individuals that develop type 1 diabetes. So that type of event has been observed. In terms of, the decision to go down the path of a beta cell versus an alpha cell, there are some alpha cell defects as well in type 1 diabetes. So I think we need to move a little bit away from this notion of this just being a beta cell disease. So there's implications that the entire pancreas is impacted. It's not just beta cells that are defective. There's certainly alpha cell defects as well.
Yeah, it's somewhat troubling because the whole pancreas is smaller, less endowed. There's impaired signaling. There's other cell types that have problems. How do you remedy that without a pancreas transplant, if that even would work? It's hard. It seems like, how do you rehab a pancreas that's in this state?
Yeah, no, there's definitely challenges. And when we think about therapies, we oftentimes think about two approaches that are needed, or a combination therapy that's needed. Certainly, one is that we have to address the underlying autoimmunity, basically the immune system recognizing beta cells as foreign, and we have to shut down that autoimmune response. But then in these individuals, if they've already developed clinical disease, then we have to remedy this deficiency of endogenous beta cell function, or potentially also think about these restorative therapies from either iPS or ES cells as well.
Well, very good. Todd, what's the best way for people to find out more about your research?
Yeah, so please check out the University of Florida Diabetes Institute websites and just Google Todd Brusko and you can find our lab and what we're working on currently. And I'd be happy to talk and hopefully address some additional questions.
That's great. Well, thank you for your work and for coming on the podcast. I appreciate it.
Absolutely. Thank you, Richard. Take care.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.